Thank you again. So it was a very, very nice talk. It makes a nice segue, I think, into this particular talk where, as you know, we are focused in stem cell therapies. I'm not going to repeat a lot of the background, but I do want to highlight that over the past several years, we've published a number of studies really investigating the molecular responses to focused ultrasound. As you saw earlier, there are a number of disease applications where these can be used. But we don't clearly understand how the ultrasound is directly interacting with the tissue to produce the molecular biological changes that we've so well characterized. Now, this is from a paper that we published last year, and this is one of the molecular responses that we identified in the skeletal muscle, and we were able to break this down into a few key checkpoints here, where immediately after HIFU, you get rises in TNF-alpha and IL-1A, and this induces expression and activation of COX-2, which then leads to the molecular responses that, in our case, we know to be critical for stem cell homing. And what we've shown through various drug studies is that by blocking any of these checkpoints, you can actually shut down the entire response, and you don't see homing to HIFU-treated tissues. But there's still a question mark here that how does HIFU generate these immediate rises in pro-inflammatory cytokines? So the first thing that we were able to do is to use COX-2 expression. This is what we found to be a good proxy for the molecular readout of our effects. And we plotted COX-2 expression versus tissue displacement and cavitation for both the kidney and in skeletal muscle. Now, I want to point out that all of these exposures are less, or generate temperature increases of less than 3 degrees Celsius. So we are working under the, operating under the assumption that these are almost entirely mechanical. And what we see here is interesting correlations in both tissues, where as we dial up pressure and displacement increases without cavitation really increasing, we are getting increases in COX-2 expression, suggesting that acoustic radiation forces alone are enough to activate this COX-2 expression. But what's also interesting is that eventually displacement maxes out. You can only push on the tissue so much, and that if we continue to increase pressure beyond then we start getting quite high levels of cavitation, and what happens is that we continue to get increased expression of COX-2. Now, we're still not quite sure if this is two distinct mechanisms at work or if there is a general mechanosensitive element in the tissue that doesn't necessarily discriminate between acoustic radiation forces specifically or mechanical cavitation forces and can be triggered by either. So the first target that we were interested in is mechanosensitive calcium channels, which Joe alluded to in his comment to the previous talk. And what we can do is give different drugs to the animal, such as gadolinium, which is a fairly non-specific cation channel blocker. We can use ruthenium red, it's slightly more specific to stretch-activated channels. And interestingly, we also tried an L-type calcium channel blocker in verapamil. And what we see is that regardless of the drugs that you use, if they're on board during the sonication, then we fail to upregulate COX-2 expression, and we fail to see enhanced stem cell homing to the treated tissues. Trying to look a little more specifically as to what this mechanosensitive receptor might be, the TRPC1 channel, the transient receptor potential C1 channel, is a good candidate. TRP channels have been shown actually previously to be opened by cavitation forces with microbubbles. And so we were able to obtain a TRPC1 knockout mouse, and we actually did the same experiment in this specific mouse where we do not see elevations in COX-2 or stem cell homing. We also did a control where we used the background strain, and we showed that both COX-2 and stem cell homing were increased to it, just not in the transgenic. So this leads us to this current hypothesis about how it works, is that focused ultrasound interacts mechanically with the cell membrane to open TRPC1 channels. Now, we also showed that L-type voltage-gated calcium channels are important, and I suspect that what's going on is you're getting local depolarization from the mechanically sensitive channel that is sufficient to activate the voltage-gated calcium channel. And together these produce a sufficient rise in intracellular calcium to activate known inflammatory pathways, and then things follow a very canonical course that leads to molecular expression and MSC homing. So again, just in conclusion, we would like to continue examining the interplay between the stretch-activated calcium channel and the voltage-gated calcium channel. We believe this being a non-thermal mechanism, that radiation forces or cavitation forces either are sufficient to induce this molecular response, and that what we see is where we are operating, so we're about an MI of about 4, we don't use microbubbles, that at that exposure settings that ARF is what actually dominates as opposed to cavitation. And the last thing would actually be to start identifying which cell types in the tissue are responding molecularly. I think that would also provide quite a bit of insight as to what's going on. So with that, thank you very much.